I'm actually going to jump in because I want to make sure that we have plenty of time for discussion. The idea here is that Nancy, Dr. Speck, and I will review the key takeaways, and then we'll hopefully end with plenty of time for Q&A. I want to remind everyone that this is a scientific key takeaways for patients, so we really try to gear this for a more general audience, not necessarily for people with scientific technical backgrounds. And I also want to highlight that we had over 19 speakers and 14 poster presentations. And so what Dr. Speck and I discussed today is not meant to be a comprehensive review of all of the presentations, which I can assure you that every single one of them had value and interesting insights. But we really aim to try to distill down some of these, key poignant points about what we learned at the scientific conference. So if you're listening as a scientist or physician that was there and presented, and we don't happen to mention your project, please don't worry. We every single presentation was highly valuable. So with that, I think I will get started. So what did we learn? Let me just move my slides here. We learned that RUNX1 FPD is not that rare, and we'll get into each of these as we go through the discussion. We also learned how germline RUNX1 mutations affect the immune system and inflammatory processes, something we've talked about many times at this annual scientific key takeaways discussion. We learned about how the number of blood stem cell clones may be lower at birth in RUNX1 FPD individuals and why that might be happening. We learned about how machine learning could be used to accelerate research by reducing time and, most importantly, cost. And then finally, we learned about how collaboration, open communication, transparency, and community prioritization can create a synergistic effect on our research progress. So we'll get into all of these, but I wanted to take a moment to make sure that we're all on the same page as we think about blood cancer development. And so in order to do this, we want to make sure that we're all using the same language and that we have a nice visual in our brains that helps us understand as we get into the key takeaways what we mean when we use specific nomenclature like clonal hematopoiesis. So with that, I'm going to show you some illustrations here. So please think of a healthy blood system as a green lawn, right? So you've got beautiful green grass, you've got the seeds of the grass, which represent the blood stem cells. And this is when the blood system is working and functioning perfectly well. In order for the blood system to work perfectly well, you can't only look at the seeds of the grass and the grass, but you have to think about what's underneath. And what's underneath is the soil. And interestingly, when I was putting together these slides a while back, I learned that there are many different components and elements in the soil. For example, air actually makes up 25% of the soil. And so I do this only to illustrate that we have to, when we think about the bone marrow and the tissue that the bone marrow resides in, we do have to think about all of the cells within the bone marrow, not just the blood stem cells, not just the immune cells, but we have to think about the osteoblasts and the adipocytes, and there are a number of different tissues there. You can also think about the soil as the entire human body as well, that the blood system is part of this very complex environment. And so this is important to keep in mind as we talk through the key takeaways. So mutations in the blood stem cells can happen, and it's a very normal process that all of us are exposed to, right? We all have mutations in a whole host in all of our tissues. And most often those mutations are not deleterious. In fact, they're deleterious to the cell that it happens to. And so that will just be, pass away, it'll die, and a green grass seed will replace it. But as we age, the soil begins to change, and the soil changes with aging through upregulation or overexpression of inflammatory proteins, inflammatory processes, and it's quite normal. And you see this as you look at bone marrow in aging individuals. And part of that process, what can happen is those mutations that happen in the seeds, the orange seeds here, a weed can grow out. And that weed is meant to represent cells that aren't really functioning the same as they would had they not had the mutation. And we call these weeds and this increase in inflammation clonal hematopoiesis. And so you can see here, these weeds are meant to represent cells that have now acquired a mutation in the bone marrow and have now behaved differently and have a different function. At the same time, you have this increase in inflammation in the soil, this change in the soil, and there's crosstalk between the weeds and the soil that can sort of drive this process along. And over time, even in the general population, what can happen is these seeds can acquire a new mutation, and that's represented by the blue and the orange seed right here. And then if these seeds begin to crowd out in the soil, you get an abundance and outgrowth of multiple different seeds. And you can see these particular seeds have a very nasty looking weed, and they start to crowd out the green grass. And so now you have a drop in blood counts. So if we're thinking about the metaphor here, the weeds are crowding out the normal healthy green stem cells, and you get a change in the number of blood cells produced, change in the number of green grass blades, and you have these like poor functioning blood system. And in the end, if blood cancer does develop, you can essentially picture that as being the entire bone marrow filled with weeds, no longer any green grass, and you have all these seeds, which are the blood stem cells that have mutations. And so the blood system can no longer function as it should. And at the same time, the soil is rich with inflammation, which is further driving this process. So what happens in RUNX1 FPD? So in RUNX1 FPD, you can imagine that an individual with a RUNX1 mutation has a relatively normal lawn, but it's just slightly different. It's blue grass instead of green grass. So we know that with RUNX1 FPD, you have lower platelet counts and you have platelets that don't function as well. But generally speaking, the blood system, you can live with a healthy blood system aside from those differences in the platelet counts and function. And the process of I want to first say that we know in RUNX1 FPD that not every individual is going to develop blood cancer. And that's really important for everyone to remember, that this is not an absolute guarantee. We know the lifetime risk is up to about 50%. And one of the really interesting, what's really interesting and important for us as a scientific community and as a community at large is to really understand what makes an individual who has a RUNX1 mutation, how are they protected from blood cancer development? Why does this process of blood cancer development not happen in them versus it happening in others? And that's an important research question. But what I want to highlight here is that there are some differences, right, between what happens in a person who doesn't have a RUNX1 mutation and a person who does. The first is the types of mutations that occur. So the clonal hematopoiesis that we see are different. So in the previous slides, I was showing it was an orange mutation and a blue and orange mutation. Well, in RUNX1, we know that there are different common co-occurring mutations as part of this process. And one you've heard us talk about in the past is BCOR, for example. So you see BCOR mutations happen early, and then you see some other mutations happen later. And the order of the mutations is also different in a RUNX1 individual versus someone who has sporadic leukemia. The other thing that's different is that it appears that the process, these factors that help drive the development of blood cancer are arising earlier in a RUNX1 individual as opposed to someone without a RUNX1 mutation. So you can see already on this slide, we have inflammation depicted by the fire happening where there's blue seeds only, right? It isn't necessarily associated with the new mutations that occur. And we also see that there's more individuals who have weed seeds, if you will, at earlier ages. So if you see on the bottom of the slide here, clonal hematopoiesis, so weed seeds, are occurring in RUNX1 individuals who are young, between ages 19 and 29, 22%. As compared to individuals who don't have a RUNX1 mutation, that rate is 0.2%. So it's very different. So there's this higher rate of clonal hematopoiesis happening. And when you look at older individuals, that difference is even greater. So you can see in individuals who are 60 or older, 7% of the general population has clonal hematopoiesis or weeds in their bone marrow. And in RUNX1, in the case of RUNX1, 83%, 60 years or older has these weeds. So this is how we believe this is happening and why there's a higher risk of leukemia. It's this sort of acceleration of these processes. Okay, so now I hope we're kind of all on the same page about how we think about blood cancer development. And I will move on to the next slide here. Okay, key takeaway number 1. RUNX1 FPD is not that rare. So Dr. Kelly Bolton from Washington University has been using the UK Biobank and other large population health databases to understand how germline predispositions to hematologic malignancies or blood cancer, like RUNX1, impact the risk of acquiring these mutations. So if you have one of these predispositions, how at risk are you for having clonal hematopoiesis? And then the follow-on question is, if you're an individual who has a germline variant like RUNX1 and clonal hematopoiesis, what is your risk of developing a blood cancer? And I won't go into the results that she shared to address those questions. Suffice it to say that there wasn't any surprises as it has to do with RUNX1. So we did see this increased risk of blood cancer as we have known now in her data set. But what was really surprising is that she identified 32 cases of RUNX1 FPD in the UK Biobank. And what that means is so in the UK Biobank, there are 450,000 individuals between the ages of 40 and 70. And so you can do a simple math calculation. It's not perfect, but you can say, okay, well, if there are 32 cases of RUNX1 in a population of 450,000, if you're to extrapolate that to the US population, which is 334,900,000 individuals, then we get an estimated risk of or rate of RUNX1 FPD cases at 23,815 individuals. And that's at minimum, because you have to remember the UK Biobank only included adults between ages 40 and 70. So it doesn't include a full population sample, and therefore the number of cases could be much higher if you had included younger individuals in that UK Biobank. And I think this is really, really important because it underscores how underdiagnosed this disease is. As you all know, we have only a little over 400 individuals in our RRP patient community. And I certainly don't think that we've identified every single diagnosed RUNX1 patient in the US, but 400 individuals is a lot less than 23,000. So I think we're a long ways off of really identifying all the individuals that have these mutations in our communities. And I think what it signals to the medical community, hopefully, is that we need to be doing a much better job of diagnosing this disease, and we need better access to genetic testing. We need a more informed medical community. We need more genetic counselors. The list goes on, but this to me, was a really striking finding. Okay, moving on. Key takeaway number 2. So in this slide, we're summarizing a number of different scientific presentations that described, how RUNX1 mutations are impacting immune cells and inflammation. And this has been a topic for the scientific community for many years now, and we continue to learn more. thanks to continued investment and effort by the scientific community to really understand this. The first key point I'd like to make is that over year after year, every time we look at results, it seems that across every single patient sample, every mouse model, every zebrafish model, every cell model, the inflammatory pathways, pro-inflammatory pathways are consistently elevated. And this is a big deal because this kind of conservation across organisms tells us that we're on the right track. In addition, or I guess to delve into the details, I should say, there were 3 independent groups at the meeting that showed that there were very different abundances of different immune cell types. So in the figure on the right, you can see one example from Dr. Esther Obeng, where you can begin to appreciate that on the left-hand panel, the bar graph is representative of a RUNX1 individual, and on the right hand is representative of a healthy volunteer control. And you can see that there are differences in the abundance of different immune cell types. So for example, there were much higher levels of helper T and B cells, as well as much higher levels of monocytes in the bone marrow. And then if you look on the right-hand side, you can see there's an abundance of Treg cells in green in the control and healthy individual, and completely absent in the RUNX1 FPD sample. So what this tells us is that we have still a lot to learn about how these differences in cell frequencies and different immune cell types may be contributing to leukemic risk. But, for example, we already know that Tregs are really important in preventing the immune system from attacking its own body. And by the way that Tregs work is they suppress autoreactive T cells. And this could be one reason why we see a much higher prevalence of autoimmune disease in individuals with RUNX1 FPD. In addition, Tregs can help keep inflammation in check. And so the loss of Tregs could be one reason for this sort of runaway inflammation that we see in the bone marrow. So lots for us to learn and dig into. And there are individuals in our community that are working to understand the T cell compartment. And so I'm sure we'll learn a lot more in the next scientific conference. Moving to the third bullet, Dr. Bresciani from Dr. Liu's lab at the NIH found that MHC class II proteins are upregulated in RUNX1 FPD stem cells. And these are proteins that help immune cells communicate with each other about an infection or an injury, and they activate the immune response. So you get this activation of the immune response when you can have antigen presentation. And so these proteins are helping to elevate that process. And interestingly, elevated levels of these MHC class II proteins have been associated with aging and the exhaustion of blood stem cells. So this may be another interesting signal or indication that the stem cell compartment is under stress. And this is one way in which the stem cell compartment is trying to address that. Here's some good news. The good news is we do have an open clinical study that tests a medicine called sirolimus, which has been shown to modulate the immune system in ways that can improve some of these deficiencies that I just mentioned above. For example, sirolimus selectively promotes the expansion of Tregs, and this has been observed in both mouse models and in human clinical studies. It's also been shown to slow the production of monocytes. So it's really important in the differentiation of monocytes and macrophages and in controlling that process. Additionally, there's evidence that sirolimus can reduce MHC class II proteins in some cell types. So sirolimus could be a very interesting tool in managing RUNX1 FPD. And hopefully many of you on this call know that there's this open clinical study. And I'll also mention that there is some evidence that imatinib may have some off-target effects on the immune system that may also play a role. But and that's a study that's also open at the NIH. Finally, there were two groups, Dr. Anupriya Agarwal and Dr. Stavrula Kustini, both identified new potential biomarkers in RUNX1-FPD, and both of these could lead to new medicines in the future. So these markers are called CD74 and SAA1, which can be measured in the bone marrow. And in a small number of patient samples, it appears that both of these increase with disease progression. So if you have an individual who has no evidence of clonal hematopoiesis, no new mutations, and you compare it also to an individual who eventually developed a blood cancer, you can see that these markers are tracking with that disease, those different disease stages. But we need further studies to make sure that this is actually true with a larger cohort. But the hope is that this could really help patients and physicians determine whether they are at increased risk or at different stages of blood cancer development. And as I mentioned, they could be new drug targets. I think that's all I'll say on this topic. And this is meant to go back to our illustration. The idea here that if you're one therapeutic approach here is to reduce inflammation, broadly speaking. So that could be reducing inflammation by controlling some of the immune cells that drive inflammation. It could be reducing inflammation by reducing certain pro-inflammatory cytokines. And so the idea would be that you would, by treating, as in this water bucket, you could eliminate inflammation and hopefully support the blue healthy RUNX1-FPD stem cells that don't have mutations at the expense of the mutated weed seeds. So with that, I will pass it on to Dr. Speck.
Hi, everyone. It's really a pleasure to see you again. I have to give you a little warning up front. I have somebody coming to install a microwave oven in 10 or 15 minutes. So I may run out in the middle, but I think I can get through this in 10 or 15 minutes. So let's go to the next slide. So one of the very interesting advances and very promising approaches came from Dr. Alex Bick's lab, who's using machine learning to advance drug discovery. So drug discovery is generally a long, expensive, and tedious process in order to identify drugs that may work. So he is trying to accelerate this process using artificial intelligence. What he's done is that if you look on the right, you see a graph that's got a little blob of gray cells and a bigger blob of red cells. What he did was he took human hematopoietic stem cells and knocked out RUNX1. And so he's got the gray cells are the normal human hematopoietic stem cells, and the red ones are the RUNX1 knocked out cells. And he compared those two cell populations to each other and then used artificial intelligence in order to determine how could you possibly perturb the RUNX1 mutant cells so that they would behave more like a normal cell. And if we could identify those perturbations that may revert the properties of the RUNX1 mutant cell to be closer to the normal cell, which ones have drugs that we could use to cause those perturbations to happen. And so basically the approach that he's using is starting with the RUNX1 mutant comparison of RUNX1 and mutant cells, using in silico predictions, using artificial intelligence to identify potential small molecules or drugs that could push the RUNX1 mutant cells to be closer to the normal blood cells. And then he orders those drugs and he tests them on the RUNX1 mutant cells to see if they actually work. So this was a very exciting development, which hopefully is very much going to accelerate drug discovery. And so the idea behind this is that if we could find a drug or a molecule that makes your your blue seeds, which are your RUNX1 mutated seeds, behave more like green seeds. I mean, it won't make them green seeds because it's not going to correct the mutation, but it may correct the properties of the blue cells to make them closer to the green cells. And by doing so, we may be able to reduce inflammation and leukemia risk. Okay, so the next, this was a really exciting insight from Vijay Sankaran's lab. And so what he has been doing is measuring how many different hematopoietic stem cells there are in normal patients compared to RUNX1 FPD patients. So he basically discovered that in patient bone marrows, there were fewer stem cell clones overall. So what do I mean by this? Well, my analogy for this is kind of like a goldfish pond. If you have a goldfish pond, let's say you're starting a goldfish pond and you populate it with a bunch of different types of goldfish, and you put them in there at a small number originally, and then they grow and expand to fill the goldfish pond. If you start with a diverse population of goldfish, you're going to end up with a diverse population of goldfish. But what we think is happening in RUNX1 FPD is that there are a smaller number of different goldfish, so smaller number of different HSCs, and that they will ultimately expand to fill the goldfish pond, but they will, in doing so, you'll have fewer different types of goldfish or hematopoietic stem cells. The next slide. So how might this happen? From a lot of embryology, historical embryology, one thing we know is that fewer hematopoietic stem cells are produced during development. So during human development, hematopoietic stem cells form at about 5 weeks of development, at a very specific time in a very small window. And we know that RUNX1 levels are really, really important for determining the efficiency of the production of hematopoietic stem cells. So in the embryo, hematopoietic stem cells are forming normally at a certain rate, but in RUNX1 FPD, we think perhaps this could be one explanation. In the embryo, that process of blood cell or hematopoietic stem cell formation is much less efficient, and so you get a fewer number born initially. Now, whatever you start with, those are going to expand to fill the niche, to fill the bone marrow, or in this case, to fill the fish pond. But in the end, then you have a much more diverse set of hematopoietic stem cells as an adult. If you don't have RUNX1 FPD, then you do if you have RUNX1 FPD. So this is one possible way that this could happen, and it's the way that I think makes the most sense based on what we know from the embryology of studies that have been done in mouse and zebrafish embryos. Next slide. Yeah, next slide, please. There's another way that this could be happening. And that could be that you start out with the same number of hematopoietic cells being made in the embryo, but you lose some of them as you mature into an adult. So this is important to understand whether the making fewer cells or losing more cells is what's going on, because we think that if you're making fewer cells in the embryo, that process is not, we will not be able to change that process, because that happens in a really small window of embryonic development before you know that fetus has even got RUNX1 FPD. On the other hand, if starting with the correct number of hematopoietic stem cells, but we're losing certain ones of them over time, that may be preventable. We don't know, but it's theoretically possible. And so one way we can start to discriminate between these two possibilities is to determine as close to birth as possible, sorry, whether you start life with fewer hematopoietic stem cells. And so this is why we would like to know if anybody has saved umbilical cord stem cells from their children. Because if we can identify in the umbilical cord that you have the correct composition of stem cells, or if you have a really reduced composition of specific cells, then that will determine really how we think about whether this could be used to treat patients as a strategy. So key takeaways number 5 are really have to do a lot with the what this foundation has really done. I mean, the collective power of the whole community has been astounding. And if you look at that group of people there, I've been in the photo, I've been in this field since the very beginning. And I could tell you that had it not been for the foundation, there would be maybe 6 members of that group of people who would be working on RUNX1 right now. And the fact that we were able to, the foundation is able to fund research has brought many incredibly impressive scientists into the field who come in with very different perspectives and technologies and expertise. And it has really grown into a large community of very collaborative people. And the meetings are so important to us because, especially when patients attend, we scientists who don't treat patients have a much better understanding of what patients are dealing with and what sorts of interventions might be tolerable to patients. I also want to point out that the foundation is working hard to generate a clinical consensus guidelines, which is going to be important for patients to be treated, across the world fairly more uniformly. And so physicians who don't encounter patients very often will have established guidelines for how to treat them. And finally, the foundation is also supporting a second biobank at UPenn, University of Pennsylvania, in addition to the NIH biobank. And the reason for this is that some patients have not enrolled in the NIH study, but we would still like to be able to collect their data and their biological material, their bone marrow samples. And in addition, Paul Liu, those of you who visited his clinic at the NIH know it's a relatively small group of people who are working on running the clinical trial and doing the biobank. And he gets many more requests for blood cell samples than he can reasonably accommodate. So having a second collection and distribution center is going to be very important for accelerating science in this area. So just to review what we've learned, Katrin, you may want to take over because I'm having a problem with coughing.
Couldn't find my unmute button. Yes, happy to. So, and thank you for being here, Dr. Speck. She's been fighting a cold the last few days, so we're lucky that she's here. So RUNX1 FPD, we talked about how it's really not that rare, and there might be 23,000 or more individuals in the US alone. So it's a much higher number of individuals that are walking around with a RUNX1 mutation than we previously appreciated. We consistently and continue to dive deeper into learning the impacts of germline RUNX1 mutations on the immune system. Really, it looks like all of the immune cell types or many of them, and all of the inflammatory processes that come with, how immune cells interact with each other. We're learning how the number of blood stem cell clones in the bone marrow may be, as Dr. Speck was just sharing, may be actually lower at birth, or they may be lost, but something is happening in terms of the diversity of the stem cell pool in the bone marrow of RUNX1 individuals. We learned about the value of applying machine learning to drug discovery and how Dr. Bick's lab is really leading the charge there, thanks to collaborations with CZI. And then finally, we're learning how valuable it is that we all come together, every single stakeholder, meaning clinicians, genetic counselors, physicians, patients, absolutely, patient family members. I mean, it takes a village for us to move the research forward. And And so the conference was a great reminder of that. And we're looking forward to hosting another combined scientific conference and patient meeting in person in 2026. So you all know that we do this, the virtual every other year and in person every other year. So in 2026, we hope to bring the entire community together, meaning patients and family members as well. So with that, I will stop and see if there's any questions or, and in fact, I can probably take it off the slideshow, or comments. Since there's several scientists and clinicians on the call, please jump in if there are things that you want to raise as important insights as well, since you know, we were not able to cover everything. Oh, did we have questions in the chat? Let's see. Nope.
I mean,
Any
I'll make
questions?
one comment, which
Sure.
is having studied serum amyloid A 40 years ago, it's very likely to be a marker of inflammation as opposed to anything else, with the possible caveat that we don't completely understand what it's doing, or at least we haven't historically, except maybe involved in lipid metabolism. So it might be feeding cells that have the receptors for it. The other comment I would make about the machine learning project is that the phenotype that he wants to correct is at the stem cell level, and maybe a lot of the pathology that we see, particularly the inflammations, is at the level of the mature cell. So it would be interesting for him to take that same compound and see if it had downstream effects, because the effects that a RUNX1 mutation might have in a granulocyte could be completely different and actually be more important in terms of pathophysiology than reverting some kind of phenotype in a stem cell. It's just a comment. That, I mean, that seems to me you'd really want to understand that.
I think that, thank you, Hugh. I think that regarding your last comment on Alex Bick's machine learning project, I think it's a valuable tool, but it's certainly not a tool that we can use in isolation. And so everything we learn from this type of machine learning tool, we'll have to validate it through in vitro models where we're using cells in a dish, and then also in vivo models, like see how does the drug that we identify behave in an entire living system. I mean, biology is complex, especially when you know, in a living organism. So your point is well taken, and I think we just have to be careful. We have to recognize the advantages of tools and what it can do, but also recognize the limitations. And but I think we could even use the tool to look at downstream cells as well, like you
Yeah,
suggest.
no, I don't disagree. The thing is that if you use granulocytes as your model cell instead of HSCs, you might come up with a completely
A
different set of
different
cells.
list. Yeah,
Yeah.
yeah.
And that's my only comment. The other comment I'd make is that almost all of the drug development process, the time and the expense, 95% of it is actually getting into the clinic. It's not the discovery part. The chemistry typically is not the rate limiting step. Knowing a good target, which this might do, in an unbiased fashion, might identify targets that we wouldn't have guessed at based on, you know, what we understand. But the real rate limiting step in drug development is the 10 years spent in the clinic.
Yeah, good point. Good point. Oh, we have some questions here. What can we do to increase the number of people being diagnosed? How do we spread the word to PCPs and hematologists? Thank you, Laura. This is, I mean, if you have any new ideas, we are open. This is something that Amanda and I and the team talk about all the time. It's one reason we do the medical education webinars that we do, and trying to work with different specialties so that we can reach different parts of the medical system. But I don't feel like we've really moved the needle yet. So any new ideas from anyone, we're all ears. All right, and then looks like we have another question. The RUNX1 mutations in the UK Biobank are all pathogenic and likely pathogenic, correct? They are all likely pathogenic and pathogenic, and those were curated by Kelly and her team. The other comment to make is that the UK Biobank is only exome, and I don't know that they can, for example, detect copy number variants. So it's also another reason why there might be a lower estimation in terms of the number of cases. So, and I think,
Yeah, go ahead.
oh, there was one more question here. How can this study be replicated in the US to provide more accurate estimates of prevalence? I mean, I think the hope is that all of us, once that grows, they're targeting 1 million, right? That we could use that as another database. But I think they're only at like 300,000 now or something. I don't quote me on that. Okay, go ahead, Hugh.
I was just going to say you know, you earlier on, you asked the question, why do some people don't get a hematologic malignancy and some do? And that's an area of research. It could well be that it is just a random process. And if you happen to get a mutation and there's no biology that you could study that would illuminate why you got a particular mutation in one place and versus. So it may be that what you want to establish is whether it is random or not,
Yeah.
as
Yeah.
opposed to what is protecting patients from getting a mutation. It seems
Yeah,
like
that's also a possibility.
yeah,
Absolutely.
yeah. It's just how you think about the problem. Yeah.
And oh, Sean, you mentioned UK Biobank is also healthy individuals. Yes, that's right. And so that also is a reason why it could be an underestimation. So I'm convinced by the data that, and we've done calculations taking studies that have looked at the rate of germline RUNX1 mutations within the population of RUNX1 mutated AML, and then tried to extrapolate from that. And we also arrive around 18 to 20,000 in the US. So it is consistent that the numbers are much greater than anticipated. Any other burning questions or comments? Happy to take comments because there are some of you who are on the call that were at the meeting.
That's a good summary. Thank you.
Thanks, Hugh. All right, I was expecting a lively discussion of questions. But that's okay. Maybe.
I mean, the only thing I would say is that there's always this issue of what's cause and what's effect. You know, if you see increased MHC class 2, it might be because, you know, there's gamma interferon in those cells, you know, and so it's really just an epiphenomenon, if you will. It's just a marker of, and yeah, and I think there's a lot of that going on in the immune system. It's hard to know, you know, like who's on first and who's on second.
Yeah.
But that's important, you know, to understand sort of the chain of communication and what's primary and what's secondary. And I don't think that's easy to know. You kind of have to do the experiment.
Yeah. Yeah, absolutely. It could be a sign of rather than a cause of. Is what you're saying.
Exactly,
Yeah.
exactly.
Yeah.
I was
Absolutely.
going to add, it was great summary. I was going to add, I was very excited to see that lots of like different labs coming to different same conclusion, like using different techniques. For example, Astor, our group, and Erica, Poly group, using single cell method, which was slightly different. Still saw changes in monocytes, immune compartment, which is very consistent, as well as HSC, Dr. Shankaran group. And then I agree with Hugh's point that it could be interferon, TNF, all those signals, because we see those also high. So it could be a marker of those signals. So
That can also upregulate those proteins, the MHC class. Yeah, absolutely.
One of the things I agree with the intriguing observation by VJ, and I kind of favor Nancy's explanation that it's a RUNX1 deficiency at the time of development of HSCs, because that fits into another potential hypothesis that those HSCs, that small number of goldfish, they need to divide much more frequently than your average HSC,
Yeah.
which might be able to snooze for 3 or 4 months before it has to divide, whereas these guys, and that is a way they could accumulate mutations in a random fashion. So it'd
Right.
be interesting to know, not only is there oligoclonality early on, but is there a higher rate of mutations in that population of cells, without having to invoke a mutagenic process at all, just mutations that occur randomly owing to division.
Right. Right. Yeah, I think I mean, so to try to translate to the lawn example, you have, you have a huge lawn of, let's say 20 by 40 feet. And you only have, in the case of a RUNX1 deficiency, you only have maybe 10 seeds that are going to have to make up all the grass, versus in a setting where you don't have a RUNX1 mutation, you have maybe 100 seeds. The number of divisions those RUNX1 deficient stem cells have to make to be able to produce, to cover the entire lawn, is so much more significant. And so therefore their exposure, or the likelihood that mutations could happen, increases because of all those cell divisions. So I think it's a very plausible mechanism of how mutations increase. And we see that, of course, when we look at the number of individuals who have clonal hematopoiesis is happening at a much younger age in individuals with RUNX1 FPD. So yeah, absolutely an important point.
I was saying those three observations all fit together nicely. Whether that's how the world works is another matter.
Yeah.
We talked a little bit about this during our small group discussions, and I think there's one version of it where you have less stem cells and they have to output more, so that means they have a higher mutagenic load, like Q saying. But I'm also curious what that means about clonal hematopoiesis detection, right? Since we have less stem cells, is it just easier to detect clonal hematopoiesis because there's less other contributors? So we have a better detection of clonal hematopoiesis in RUNX FPD, but are there necessarily more clonal hematopoiesis? Is it just the denominator, the number of kind of normal non-mutated ones are just fewer? And I think we should, I know we have a higher rate to leukemia development, but we really have to ask ourselves, is higher clonal hematopoiesis actually really higher risk in the exact same way? Or is the denominator, just the amount that you're dividing against, just really smaller? And it kind of lends some
question.
Yeah.
Really great question. I'm always interested in the denominator.
Mm-hmm. Yeah, that's a really good point.
Well, Bobby, how do you sort that out? That's
yeah, it makes you wonder if one way to think about it is we've obviously started to nail down some potential mechanisms for why we have less clones, right? And maybe what the stresses are that are selecting for specific ones. But maybe the way to flip the question on its head is, how do you promote the fitness of other clones, right? Or create more polyclonality. So maybe the cells to target are not the ones that are expanding, but perhaps dormant or quiescent ones that we're kind of maybe more blind to, right? That would be the positive outcome to it, is can we just increase the denominator, right?
Yeah.
And
So like
maybe
push the sleeper cells to start contributing.
yeah, and this is a field I'm not super familiar with, so this is a bit of scientific speculation, right? So I don't want to run wild with it. But are there approaches from our colleagues that work in regenerative medicine that should be looked at here, right? In the context of boosting hematopoietic stem cell, not necessarily output, but taking maybe the most dormant ones that we don't maybe think are contributing and getting them to contribute. Maybe you can kind of bathe down the clonal hematopoiesis load.
Well, that suggests two things in my mind. One is it may be that the wild type cells are more sensitive to the inflammatory environment. So if we control that, those more dormant cells or cells that are really held back are have a chance to flourish. And the other thing is that I hate to go back to this, but LSD1 inhibitors cause an expansion of stem cells. And this is most evident, in the use of UM171 ex vivo. It's toxic in vivo, but there are certainly other LSD1 inhibitors that are not toxic that have shown to expand wild-type stem cell populations in vivo, and that may be a strategy actually.
Mhm.
Which anybody who wants some of that, I've got plenty of that.
Let's test that. Oh, and we have a question from Kay on Dr. David Young. Yes, Dr. Young presented a clinical trial that he's a protocol that he's drafted to potentially test a JAK2 inhibitor called pacritinib. I don't know, where he is with whether he's going to the IRB and getting approval. I don't know where that sits, but I will get back to you because I'm going to reach out to him and speak more about that. I will say that when Dr. Young presented, there was discussion about, given that the RUNX1 community at the moment that's involved is very small, how will this community be able to push forward some of these clinical studies that are now launched? So Dr. Cunningham's on the call and she's running this imatinib clinical study at the NIH. Dr. DiNardo, who's at MD Anderson in collaboration with Dr. Agarwal, who's also here on the call, is running a sirolimus study. So, you can imagine if we have such a small patient pool, how are we going to advance these studies and learn from these studies if we have, too many clinical studies all open all at once, because then we're in a situation where none of them advance and then we learn nothing, and that would be terrible. So that did come up as part of the discussion, just so that you're all aware of that.
I was hoping to add like few points for the stem cell conversation, like HSC
Yeah.
pool. So it's a smaller pool for sure. But what we noticed that one point what Hugh mentioned that healthy cells are more exhausted under inflammatory stress, and they self-renew slower compared to FPD. So it's a smaller pool, but it retain longer. They divide slow. So I feel like somewhat they are acting as a little bit dormant. Like we didn't dissect within HSC pool, which is HSC pool doing, or are there two different pools? But I think smaller pool still survive longer, and that we see in multiple assays. So maybe that's the reason that contribute to clonal hematopoiesis or acquisition of more mutation early on, like things like that.
When you say smaller pool aneupria, are you talking about the actual number of hematopoietic stem cells, or are you talking about the clonality, like the diversity of
I'm not talking about clonality, like it's total cell number.
total cell number? Oh, and so you see, like, is it a statistically significant difference between?
I was going to say it's not a huge difference, right? In the number, like we were talking about denominator earlier.
Yeah.
I was saying it's not a huge difference in my hand, and I think clinical study also show that. So it's not very huge difference, but it's lower at the lower end.
Got it. Okay. Any other comments, questions, suggestions, ideas? All right. Well, thank you, everyone. I hope you enjoy the rest of your Friday and have a wonderful weekend. And thank you so much for